This is week 3, Principles of Clinical Cytogenetics and Genome Analysis, Part 1. The study of chromosomes, their structure, and their inheritance is called cytogenetics. The science of human cytogenetics dates back to 1956 when it was first established that the normal human chromosome number was 46. Here we have a slide showing you what the chromosomes look like in 1882 and what they can now be seen as with the specific banding patterns. If we look at the portrait of a chromosome, we see that a chromosome consists primarily of DNA and proteins with a small amount of RNA. The different chromosome types have long been described and distinguished by size and shape using stains and dyes to show the darkly staining area, which is the heterochromatin, which consists mostly of repetitive DNA, and the lighter staining areas, which is the euchromatin. And this contains most of the protein encoding genes. The largest constriction of the chromosome and where the spindle fibers attach are the centromeres. Bases that form the centromere are actually repeats of a 171 base DNA sequence. It's replicated at the end of the S phase. It's facilitated by a centromere protein A, and this protein is passed to the next generation and is actually an example of an epigenetic change. Telomeres are chromosome tips, and they're composed of many repeats of TTA, GGG. They shorten with each cell division, and each telomere repeats the sequence. I'd like you to visit this website to read an article about telomeres. I'd also like you to visit this website to complete an activity on telomeres. If we look at this chromosome, we can see that the shorter upper arms here are called the P arms. We have the telomeres here at the end. We have the heterochromatin, and we also have the centromere. So the P arm is called P arm because it is French for petite. The long arm, or the Q arm, is the longer arm. And here you can see some euchromatin. And again, you see the telomeres. Subtelomeres, where the chromosome is the chromosome region between the centromere and the telomeres, consists of 8,000 to 300,000 bases. Near the telomere, the repeats are similar to the telomere sequence, and it contains at least 500 protein encoding genes, which is about 50% are multi-gene families that include pseudogenes. Here is the centromere positions. This is very important for you to start to learn this so that you will be able to look at the karyotypes and to be able to interpret them. If we're looking here and to learn the different chromosome shapes. If we look at the telocentric, we see that the centromere is at the tip. For acrocentric, it is close to the center. For submetacentric, it's off center. And for metacentric, it is in the middle. Karyotypes are a chromosome chart. It is a major clinical tool. It displays chromosomes arranged by size and structure. Humans have 24 chromosome types. The autosomes are numbered 1 to 22 by size and are the same in both males and females. The sex chromosomes are Y and X. Here is an example of a normal male, 46 XY male, and the chromosomes have been stained with what's called G banding or GEN600 banding in this specific one. Karyotypes are useful at several levels. They can confirm a clinical diagnosis, they can reveal effects of environmental toxins, and they can clarify evolutionary relationships. In order to visualize chromosomes need to be extracted, and then they need to be stained with a combination of dyes and DNA probes. Initially, dyes were used to stain chromosomes a uniform color. They were grouped into decreasing size classes, and these were designated A through G. In 1959, the first abnormalities were seen with Down syndrome, Turner syndrome, and Klinefelter syndrome. Only large deletions and duplications could be seen at this time. Improved staining techniques gave banding patterns unique to each chromosome. These stains specific for heterochromatin, which is dark staining, and a band represents at least 5 to 10 million DNA bases. Researchers also found that by synchronizing the cell cycle of cultured cells, they can reveal even more bands per chromosome. Fluorescence in situ hybridization, FISH, was introduced, and with this we had the ability to highlight individual genes. It's more targeted since it uses DNA probes. The probes are attached to molecules that fluoresce when illuminated, producing a flash of color precisely where the probe binds to a complementary DNA sequence among the chromosomes of the patient's sample. Here is an example of FISH. DNA probes are labeled with a fluorescing dye, and these bind to the complementary DNA. And as you can see, there are fluorescent dots, and these fluorescent dots correspond to 3 copies of chromosome 21. This reveals the extra chromosome 21. With FISH, you can paint entire karyotypes by probing each chromosome with several different fluorescent molecules. A computer then integrates the images and creates a unique false color for each chromosome. Many laboratories that perform amniocentesis or CVS use FISH probes for chromosomes 13, 18, 21, X and Y to quickly identify the most common problems. The previous slide that we looked at demonstrated trisomy 21, where we saw the 3 number 21 chromosomes. In looking at chromosome abnormalities, a karyotype may be abnormal in chromosome number, in chromosome structure, and abnormal chromosomes account for at least 50% of spontaneous abortions. Some of the clinical consequences of abnormalities and variation in chromosome structure and mechanics are that you can have birth defects and developmental disorders, Roberts syndrome, premature ovarian failure, telomere syndromes, and effects in longevity, as well as you can have congenital abnormalities in inherited defects in genes encoding the key components of the mitotic checkpoints at the kinetochore. In looking at the term aneuploidy, the normal chromosomal number is euploid. Cells with extra or missing chromosomes are aneuploid. Most autosomal aneuploids are spontaneously aborted. Those born are more likely to have an extra chromosome, trisomy, rather than a missing one, monosomy. Non-disjunction. The mitotic error that causes aneuploidy produces a gamete with an extra chromosome and another one missing a chromosome. Non-disjunction during meiotic division number 1 results in copies of both homologs in one gamete. Non-disjunction during meiotic division number 2 results in both sister chromatids in one gamete. This is a slide showing non-disjunction at meiotic division number 1. If you look down here at the gametes, these gametes can result in zygotes that would be trisomic or monosomic. This is a slide showing non-disjunction at meiotic division number 2. And in this case, the gametes would result in having 2 zygotes that have the normal number, euploid, one that is monosomic, and one that is trisomic. This is a slide showing the maternal age effect on trisomy 21, which is Down syndrome, which is showing that as a woman gets older, she has an increased chance of having a child with Down syndrome due to non-disjunction. Polyploidy. This is where a cell has extra chromosome sets. Could be triploid, 3N. Cells have 3 sets of chromosomes. This is produced by fertilization of 1 egg by 2 sperm, fusion of a haploid and diploid gametes, and it accounts for 17% of all spontaneous abortions and 3% of stillbirths and newborn. And here you have a karyotype which is illustrating triploidy, where you see 3 of each chromosome. A mosaic arises during mitosis, producing groups of somatic cells with the extra or missing chromosomes. An individual with 2 chromosomally distinct cell populations is called a mosaic. Mitotic non-disjunction event is what happens early in development, and this can have a serious effect on the health of the individual. In looking at trisomies, many autosomal aneuploids cease developing as embryos or fetuses. Frequently seen, however, are trisomies in newborns that have extra number 21 or number 18 or number 13. And these chromosomes carry fewer genes than other autosomes. In this table, we can see a comparison of Pateau syndrome, which is trisomy 13, Edwards syndrome, trisomy 18, and Down syndrome, trisomy 21. We can see the incidence at birth at which Down syndrome has the greatest incidence, and the percent of conceptions that survive 1 year after birth. And Down syndrome is, again, the leader into this category. In looking at spontaneous abortions, most of the 50% of spontaneous abortions that result from extra or missing chromosomes are 45X, triploids, or trisomy 16. About 9% are trisomy 13, 18, or 21. More than 95% of newborns with atypical chromosome numbers have an extra chromosome 13, 18, or 21, or an extra or missing X or Y chromosome. These conditions are rare at birth and affect only 0.1% of all children, but non-disjunction occurs in 5% of recognized pregnancies. This is a picture of a girl that has trisomy 21, Down syndrome. It is the most common trisomy among newborns. There are distinctive facial and physical problems, and I will be discussing this in the next module. Here is a karyotype which has been banded and is showing you the 21, number 21 chromosome. This is trisomy 21, so it is 47 XX with that extra 21. Trisomy 18 or Edwards syndrome is due to non-disjunction in meiotic division number 2 in the oocyte, and generally these babies do not survive. They have serious mental and physical disabilities. They have a distinctive feature. They have oddly clenched fists. Trisomy 13 Pateau syndrome, very rare, and generally these babies do not survive 6 months. They have serious mental and physical disabilities, and one of their distinctive features is their eye fusion, where they may have no eyes, they may have just one eye. This is a slide showing how non-disjunction can lead to sex chromosome aneuploids. Here we have a normal male, 46XY, a normal female, 46XX. The consequences of female non-disjunction are that babies could be born 47XXY Klinefelters, 47XXX triplo X, 45Y, which is non-viable, 45X, which is Turner syndrome, or in the case of male non-disjunction, we would get different results if the non-disjunction took place in meiotic division number 1 or meiotic division number 2. In the case of meiotic division number 1, you could get 45X Turner syndrome or 47XXY Klinefelter syndrome. If it took place in meiosis number 2, then you would have babies that could have 47XXX triplo X, 47XYY Jacob syndrome, or 45X Turner syndrome. If male and female non-disjunction took place, the result could be 48XXYY syndrome. If we look at 45X syndrome, Turner syndrome, we see that it occurs 1 in every 2,500 female births. 99% of affected fetuses die in utero. Some of the features are short stature, webbing at the back of the neck, incomplete sexual development, which results in the child being infertile, impaired hearing, and individuals who are mosaics may have children. The Triplo X syndrome is 1 in 1,000 female births. Few modest effects on phenotype, and these include tallness, menstrual irregularities, and slight impact on intelligence. The X inactivation of two X chromosomes occurs, and cells have two bar bodies. It may compensate for the presence of an extra X. Klinefelter XXY syndrome, 1 in 500 male births. Phenotypes include incomplete sexual development, rudimentary testes and prostate, long limbs, large hands and feet, some breast tissue development, common cause of male infertility. XXYY syndrome arises due to non-disjunction in the oocyte and sperm. It's associated with more severe behavioral problems than Klinefelter syndrome. ADD, obsessive-compulsive disorder, and learning disabilities are associated with this syndrome. Individuals are fertile, and they can be treated with testosterone. Jacob syndrome, XYY. One male in 1,000 has an extra Y chromosome. This occurs because of non-disjunction in the male. 96% are phenotypically normal. They have modest phenotypes, great height, acne, speech and reading disabilities. Studies suggested an increase in aggressive behaviors. However, these are not supported. And in fact, if you read this article here at this website, you will read about how people were misdiagnosed with Jacob syndrome, including the mass murderer Richard Speck.